Hello, everyone. My name is Alejandro and I'm here with Rohith, and today we are going to talk about high concentration antibody formulations and their application towards monoclonal antibody therapeutics. So, before we discuss what high concentration antibody formulations are, the first thing we're going to talk about is what a monoclonal antibody is, which they are usually used as a therapeutic, and they are synthesized by a population of identical B cells that are grown in a cell culture, and these antibodies are homogeneous and all recognize the same epitope. They mainly function as therapeutics, as I stated, and they have merged as a major class of biopharmaceuticals for treating various diseases that include cancer, different autoimmune disorders, and infectious diseases. And as of between 1996 and 2022, there have been about 130 FDA approved monoclonal antibodies, according to the Antibody Society, and they are estimated to be a $221.7 billion industry as of 2023, with a prediction to increase to $392.6 billion by 2032. And so there are two different types of administrations for monoclonal antibodies. One is subcutaneously, and another is intravenously. And recently, there has been a push towards a subcutaneous administration. This is just due to the shorter injection times and the ability for patients to self-administer at home. And for subcutaneous injections, it is required that you have a high dosage of monoclonal antibodies, as well as you have to take into consideration the restricted dosage volume that is required for these injections. And so in order for this to become a viable option, we would have to use high concentration antibody formulations. So now I'm going to discuss how an antibody formulation is considered a high concentration antibody formulation, as well as what are the advantages of this type of formulation. So for a formulation to be considered high concentration, the concentration of antibodies must be more than 50 milligrams per milliliter. And so the motivation to develop these types of antibodies is, one, we would want to use it for subcutaneous injection, but also it would improve the patient compliance and convenience. And this is because these types of formulations enable the administration of high doses in smaller injection volumes, which allows for frequent injections and shorter administration times. And so patients are more likely to adhere to the treatment regimens that are more convenient and less painful. Another motivation is to reduce the healthcare costs for both the patient and faculty. So self-administration and less frequent dosing can lead to reduced healthcare costs. There can also be fewer hospital visits and less time that healthcare professionals require in order to administer these, and this can result in a significant cost savings for healthcare systems and patients. And the third motivation is that they have an enhanced therapeutic efficacy, where high concentration formulations can enable the delivery of higher doses, and higher doses can lead to an improved therapeutic outcome in some cases. This is particularly important for diseases that require high antibody concentrations to be an effective form of treatment.
So there are challenges of high concentration antibody formulations. So the first challenge would be increased viscosity, and it is due to the protein-protein interactions. So the attractive forces between protein molecules, such as hydrophobic interactions and hydrogen bonding, can lead to the formation of transient networks that increases viscosity. And it is also due to pH and ionic strength. So the viscosity of antibody solutions is typically lowest near the isoelectric point of the protein, where the net charge is zero, and it increases as the pH moves away from the isoelectric point due to increased electrostatic repulsions. And high ionic strength can shield electrostatic repulsions between protein molecules, leading to increased viscosity. It also depends on excipients. Some excipients such as sugars and polyols can increase the viscosity of formulation by preferentially excluding protein molecules and increasing their effective concentration. However, there are other excipients like amino acids and salts that decreases the viscosity by disrupting protein-protein interactions. And high viscous can affect the manufacturability of the formulation by increasing the pressure required for pumping, filtration, and filling operations, and that leads to longer processing times and potential equipment damage. It may require high forces to fill the syringe, leading to user fatigue and incomplete dosing, and it can also result in increased injection times, pain, and discomfort for the patient. And it can also result as a risk of needle clogging or breakage. So the other challenge of high concentration antibody formulation is stability and aggregation, and it is due to the reduced conformational stability. Like high antibody concentrations can lead to increased molecular crowding and excluded volume effects, which can alter the conformational stability of the protein. Marginal conformational stability can expose hydrophobic regions or aggregation prone sequences, promoting intermolecular interactions and aggregation. And it can also be due to limited solubility. As the antibody concentration increases, the solubility limit of protein may be reached, leading to phase separation and precipitation. And these precipitated antibodies can form irreversible aggregates, compromising the stability and quality of the formulation. And the presence of these aggregates in high concentration antibody formulations can have several detrimental effects, such as reduced efficacy. Aggregates may have altered binding affinity or biological activity compared to the native monomer, leading to decreased therapeutic efficacy and increased immunogenicity. Aggregates can elicit an immune response in patients, resulting in production of anti-drug antibodies that neutralize the therapeutic effect or cause adverse reactions. And they can shorten shelf life. Aggregation can accelerate during storage, which leads to the product instability and reduced expiration dates. So there are two major dosage forms of protein formulations, like one is liquid formulations and one is freeze-dried solid. So there are a couple of advantages of liquid formulations over freeze-dried solids, which includes it's ready to use, which eliminates the risk of errors during reconstitution, and it is easily administered through intravenous, subcutaneous, and intramuscular injections. And liquid formulations have simpler manufacturing processes compared to lyophilized formulations, which require additional steps like freezing and drying. So, lower manufacturing costs and reduce the risk of contamination. So, there are key considerations for liquid formulations such as stability, aggregation, and degradation. So, antibodies are prone to various degradation pathways such as deamidation, oxidation, and hydrolysis, which can compromise their structural integrity and biological activity. For example, deamidation of asparagine and glutamine residues. So, it occurs through the hydrolysis of side chain amide group, leading to the formation of aspartate and isoaspartate. So, deamidation can alter the charge distribution and stability of antibodies. So, and there are other factors as well, such as pH, temperature, sequence context that influence the rate of deamidation, which again influences the stability of the liquid formulations.
Oh, so the second method for high concentration antibody formulations is a crystalline monoclonal antibody suspensions. And these crystalline monoclonal antibodies are typically produced by a freeze drying method, as Rohith had mentioned earlier. And the suspensions that are used are typically an organic solvent. However, they have displayed cases of being slightly toxic to humans. So there has been a push to use hydrogels as the suspension, as they don't show any signs of toxicity within humans. These types of formulations are ideal for subcutaneous injections. And this is mainly due to one, they have a reduced viscosity, which is necessary. They also have a high stability within the suspension and immobilized state, and this actually leads into them ending up having a higher shelf stability. And then they also have a sustained release of the therapeutic, which means that when you get the subcutaneous injections, there is now a longer time between each injection that you have to take, just because you have that sustained release that will keep the therapeutic within the body a lot longer. However, there are some drawbacks to this method of formulation. The first one is that there is a slow absorption of monoclonal antibodies within the patient's body. They also take a long time to circulate throughout the blood system. And then again, as I mentioned earlier, there is a potential for toxicity from the suspensions being used. And then also During the manufacturing process, there is the potential for these crystals to settle at the bottom of the suspension, which you wouldn't want because in order for this to be as effective as possible, you would want to have your crystals evenly distributed throughout the suspension. So to conclude our presentation, we would just like to make the next few statements. With one, we have noticed that there has been an increase in the need for new monoclonal antibody therapeutics, especially with the drive towards having them administered subcutaneously. And in order to achieve this type of administration, we would need to develop high concentration antibody formulations, which would have a low viscosity, high protein stability, and a long shelf stability are necessary. And so we discussed the two different types of methods for this type of formulation, and that includes liquid formulations and crystalline monoclonal antibody suspensions, and we have discussed their advantages and disadvantages. And so we would like to conclude with the statement that high concentration antibody formulations have a potential to impact the healthcare and biopharmaceutical industries. And thank you for your time.